The action potential arises from the coordinated activation of two conductances, a sodium conductance that activates rapidly and drives the rising phase of the action potential, and a potassium conductance that activates more slowly and contributes to the falling phase of the action potential and the undershoot. How can we learn more about the behavior of these conductances? One difficulty in studying the behavior of these conductances is that they're changing over time, rising and falling as the action potential proceeds. They also change with voltage. The increased activation of the sodium conductance with depolarization causes the regenerative rise of the action potential at voltages more positive than threshold. Analyses could be made much easier if one of these elements, time or voltage, could be controlled. The voltage clamp technique does just that. It controls or clamps the voltage across the membrane to whatever value an investigator chooses, so that the changes in sodium and potassium conductance that occur with time can be studied. Let's examine how voltage clamp works. First, the neuron to be studied, in this case a squid giant axon, is placed in a dish containing saline solution. The membrane voltage, Vm, is measured by an amplifier using an internal recording electrode inserted into the axon, and an external reference or bath electrode. The command voltage, Vc, is the desired membrane voltage and is set by the investigator. The difference between Vm and Vc is determined by a comparator. If Vm is not equal to Vc, this comparator generates a difference signal. This difference signal is used by the voltage clamp amplifier to generate a current that's injected into the axon via the current passing electrode in order to make Vm equal to Vc. This feedback circuit keeps the membrane voltage as close to the command voltage as possible and operates virtually instantaneously. Finally, the amount of current required to keep Vm equal to Vc, the injected current, can be measured and recorded. This means that as voltage-gated ion channels open and close over time in response to changes in Vm imposed by the voltage clamp, the current generated by these channels can be recorded and analyzed. Let's try some simple voltage clamp experiments. First, what happens if the membrane voltage and the command voltage are identical? The arrow indicates the start of the experiment, that is, when Vc is set at -65 millivolts. Since Vm and Vc are already identical and membrane currents are at their resting steady state, there is no change in voltage. Therefore, no current is injected into the axon to oppose any changes in membrane currents. Now, what happens when we impose a new voltage on the membrane by setting Vc to 0 millivolts? When the membrane potential is stepped to 0 millivolts, a very brief capacitive current is seen, followed by a characteristic pattern of inward and outward currents. The capacitive current occurs because the step from one potential to another alters the charge separation and thus the electrical potential difference across the membrane. Once the new potential is reached, there's no more capacitive current. Initially, Vc is at the holding voltage, -65 millivolts, which is identical to the resting potential. When Vc is adjusted to 0 millivolts, the voltage clamp amplifier injects current to push the membrane potential to 0 millivolts. Now, different types of ion channels in the axon membrane open and close, such that one sees an initial surge of inward current followed by a more slowly developing outward current, the late or delayed outward current. The transient inward and delayed outward phases of the current trace recorded after the command voltage was set to 0 millivolts correspond to the sodium and potassium conductances measured during an action potential. Let's explore why this is so. In fact, the inward and outward currents are separate. One of them, the early current, is carried by sodium ions and is thus called sodium current, INA. Sodium channels are said to be voltage-gated because they activate rapidly in response to depolarization. Depolarization not only causes the sodium conductance to activate, but also causes it to decrease over time or inactivate. The other current is carried by potassium ions and is called the potassium current. Voltage-gated potassium channels activate more slowly than sodium channels and do not show inactivation in axons. How do we know which ions carry these inward and outward currents? Specific blocking agents can be used in voltage clamp studies to dissect out the currents that underlie the action potential. Tetrodotoxin, TTX, a toxin found in pufferfish and in the skin of certain frogs and salamanders, specifically blocks sodium channels. In the presence of tetrodotoxin, only potassium channels open during the voltage clamp depolarization. These potassium channels can thus be studied in isolation. Tetraethylammonium, TEA, a large organic cation, specifically blocks potassium channels and therefore allows us to isolate the sodium current. This permits examination of the detailed properties of sodium channel activation and inactivation. Since the sodium current is an inward current dependent on extracellular sodium concentration, it can be eliminated by removing all external sodium. Under this condition, the recorded current is similar to the current seen in the presence of TTX, which blocks sodium channels, with one major difference. There is an initial outward surge. What is this current? Remember that even though there is no external sodium, there is still sodium present inside the axon. This sodium can flow outward down its concentration gradient through the open sodium channels.